You've probably heard of cell membranes, but now you're actually going to know what they're made of. And they're mainly composed of phospholipids, this thing you see right here, which have a hydrophilic polar head. Hydrophilic meaning it can interact with water. And why? Because it's polar, and water is dipolar. Polar can interact with polar. Okay? This is why, as you'll see in the diagram later, these are going to be facing the water in the bilayer. Okay? Now, what about the tails that you see right here? So as you should know, tails that are straight, like this, fatty acids which are straight, are saturated, while those that have a kink or a band are unsaturated. Okay? Now, monounsaturated, we're going to have less kinks than polyunsaturated, but that's for another topic. Now, both of these tails are hydrophobic. Phobic, they're not literally, but scared of water. They don't interact with the water. They're going to be inside the membrane, as you'll see later. Why? Because they're non-polar, and water is dipolar. Now, as you can see, because the phospholipid has both hydrophobic and hydrophilic regions, it is something that we call amphipathic. Let's have a look into more detail at the hydrophilic polar head. The reason that the phospholipid's head, polar head, hydrophilic head, is polar is because of the charged or polar functional group, also known as the R group, that it has. Okay. Now it also has a 3-carbon backbone of glycerol. This thing right here, 3-carbon backbone of glycerol. Notice how all of these have a carbon, and then it has a negatively charged phosphate group. Okay, so this would be the phosphate group, and it's negatively charged. Now, as I've said before, the head is hydrophilic, it's polar, and the tails are hydrophobic, non-polar. This means that the heads will be facing out and the tails will be inside. Okay, so the heads interact with the water and with the cytoplasm, and the tails are inside because they're scared of the water. Okay, obviously they're not scared of the water, it's just a way to explain it. Personification. Okay, now moving on to what things can pass or not pass this cell membrane, let's look at steroids, oxygen, and CO2 because they are hydrophobic and non-polar. These molecules, which are hydrophobic and non-polar, can actually pass through the bilayer. They are permeable. That's just a fact you need to remember. Moving on. Now, ions such as sodium, chlorine, potassium. Okay, ions will always have a positive or negative symbol. Okay, and they'll be elements of the periodic table. These are impermeable. They cannot pass through the membrane unless there's a channel protein and transport protein, which we'll look at later. But through the layer of simply phospholipids, they will not pass. They shall not pass. Now, tiny polar uncharged molecules are semi-permeable. Okay, water is polar, urea is also polar, and they are uncharged. Okay, this is the thing. If they're charged like ions, they're impermeable. And lastly, large polar and uncharged molecules, okay, such as glucose or sucrose, are mostly impermeable. This is just something that you need to remember. Okay, so the worst thing we can have is a charged molecule. Okay, and then a polar uncharged molecule is semi-permeable because it's tiny in the case of water and urea. But if it's large, like glucose or sucrose, it's mostly impermeable. Simple diffusion. This is the movement of molecules from a high concentration gradient to a low concentration gradient. In other words, that the movement of molecules is down the concentration gradient. And this eventually results in equilibrium or balance. Okay? An example of this, the movement of non-polar molecules such as oxygen and CO2 transported by red blood cells, also known as erythrocytes, from capillaries to cells in and out respectively, like in this diagram. Okay? So as you can see here, this shape right here is the blood capillary, and the CO2 is going into the alveolus, okay, in around your lungs. Or for example, here, the red blood cell is supplying oxygen to the tissue cells, and the tissue cells are exchanging it for CO2, which is bringing back into the red blood cell. Okay, again, this links to other topics. Let's have a look at integral proteins, okay? So have in mind that hydrophobic interacts with hydrophobic, or nonpolar interacts with nonpolar. These integral proteins have hydrophobic regions. This is why they're embedded in the membrane protein, okay? Remember that inside the membrane protein are the tails, the fatty acid tails, which are also hydrophobic. And this is why it has regions inside of the membrane. But they're amphipathic, so they also have hydrophilic regions. And hydrophilic interacts with hydrophilic. And because of that, they also interact with the extracellular fluid. Okay, this is going to be more clear when we look at a diagram. Okay, another type of protein, peripheral proteins. They are hydrophilic in nature, and they do not have any hydrophobic regions. If they're hydrophilic, they're going to interact with the water. They're going to be polar. Okay, so they're going to have interactions with the extracellular fluid. Okay, and because they don't have any hydrophobic regions, they're not going to interact with the fatty acids inside the membrane, meaning that they're only on the surface of the membrane. Now, before we look at transport proteins, let's have a reminder on peripheral proteins. Okay, so as you can see, this peripheral protein right here is only on the surface. Why? Because it only has hydrophilic regions. Whereas the integral protein does interact with the surface because it has hydrophilic regions, but it also interacts with the inside of the membrane, the fatty acid tails, because it has hydrophobic regions. Now finally, moving on to transport proteins, these transport or facilitate the movement of molecules, obviously, and we're going to look at two types. Channel proteins, they form like a tube, okay? This right here would be a channel protein, and carrier proteins, which we'll look at later as well. Now these are transmembrane proteins, if we're looking at channel proteins, that form tubes or channels, as I said before. Transmembrane basically means that they move through the whole membrane, as you can see. Well, that's not a channel protein, sorry. They move through the whole membrane, okay? As always, any questions can go in the comments. I'm not too convinced about my explanation there. There's going to be other types of proteins in the membrane or membrane proteins, but all of them are going to have certain functions such as cell-to-cell recognition, acting as receptors for chemical signals, okay, binding sites for hormones, neurotransmitters that trigger certain responses, and they can also act as enzymes, okay? So as I said before, the channel and carrier proteins are for transport. Then there's integral proteins containing glycolipids or glycoproteins, which we'll look at later, for recognition. Then some of them have like these active sites for acting as a binding site or receptor, and some can act as enzymes. Aquaporins are a type of integral protein, okay? An example of an aquaporin is a tetrameric protein, which is made of 4 monomeric subunits, these 4 that you can see on the left side of your screen. Now, these contain water channels, these red circles right here, that are lined with specific hydrophilic side chains. So on the sides, there's like these chains which are hydrophilic, meaning that they interact with water. Okay, so just look at the name aqua and don't tell me that this isn't literally screaming that their function is to allow the passage of water. Okay, they don't allow the passage of ions, but they allow the passage of water. Now, kidneys reabsorb water. Okay, and this means that they need a higher concentration of aquaporins, because the more water, the more aquaporins you're going to need, because the more water you want to absorb or reabsorb. Okay, so basically their function is to allow water to go in, and that's why their water channels have these hydrophilic side chains. We've looked at simple diffusion. Now let's look at facilitated diffusion. Okay, facilitated, let me underline it, is a key word. So it's going to be the usual movement of molecules down the concentration gradient from high to low, but this time it's going to be assisted or facilitated, as the name suggests, by a transport protein. Okay, such as a channel protein. Now, we saw before that ions are charged, okay, and charged molecules cannot go through the phospholipid bilayer. It can go through the head and the tail. It has to go through a special protein called a channel protein. Okay, so these channel proteins, sorry, can select. They have selectivity of which ions go in and which ions go out or can't go in or out. Okay, now this is thanks to the binding sites of the hydrophilic amino acid side chains that they have. Okay, so these chains on the sides, which are hydrophilic, have binding sites to detect certain ions, okay, and either let them pass or not let them pass. And they also have that the size of their pore can act as a filter. So just make sure you note that down, because it's a bit hard to remember. So imagine that this is your channel protein. What are the stimuli that can open or close it? Well, first one, changes in voltage, okay, in, you could say, electricity, and these are those that respond to this voltage are called voltage gated protein channels. Another one, the binding of small molecules, okay, such as oxygen. When this binds, it can either open or close the channel. Okay, this means that it's a ligand gated protein channel. All right, and last one, mechanical forces such as pressure, force. These can also stimulate the opening or closing of the channel. We've looked at channel proteins a lot. Let's look at carrier proteins for once, okay? So, carrier proteins are specific to a certain molecule. In the case of glucose, GLUT is going to be the carrier protein for it. Boom. Glucose binds to it and the carrier protein helps it go down, okay? So when red blood cells need glucose, what's going to happen? The GLUT is going to carry it to them, delivered. Molecules can move into cells through simple diffusion, through the phospholipids, okay, if they're permeable, semi-impermeable, or even mostly impermeable, okay, and the molecules which are charged and polar, such as ions, which can go through simple diffusion, go through facilitated diffusion, okay, from the outside to the inside through the channel proteins and carrier proteins. But what about moving molecules from the inside of the cell to the outside? Okay, because in this case we're moving from a low concentration gradient to a high concentration gradient, which is the opposite of simple diffusion. So we can't do simple or facilitated diffusion in this case. Sorry. And that's when active transport comes into play. Now it does require something called protein pumps, which need the hydrolysis of ATP to ADP. Basically, they need energy, okay? This exergonic reaction that uses up energy. No, it releases energy, sorry. So we use this energy to move these molecules out of the cell. And there's two types of active transport. The direct that we've just seen, that is by exergonic reactions, okay? They're called ATPases or ATPase pumps, which are responsible for this. And there's also a co-transport or an indirect active transport, which is that when one of the solutes, okay, when one of these moves, it's going to drive the others to move as well, okay? So when it moves in this direction, then it's like an attraction, right? It's like a, how do I say it? Like it drags the others with it, okay? And that would be it. Quick activity, I want you to pause the video and tell me active transport, which of these four is it? Simple diffusion, which of these four is it? Okay, and so which one goes with which? Pause the video right now and I'll give you the answers in 3, 2, 1, let's go. Okay, so here are the final answers. Simple diffusion goes from a high concentration gradient to a low concentration gradient inside the cytoplasm, and it can go through the phospholipids. Facilitated diffusion using channel proteins. This right here is a channel protein. It goes from a high concentration gradient to a low concentration gradient. Facilitated diffusion using a carrier protein. Again, goes from high to low, and the carrier protein is gonna, well, carry it, transport it. And lastly, active transport goes from a supposedly low concentration gradient to a high concentration gradient using ATP, the exergonic reaction, using energy. Perfect. Last thing, the selective permeability, so the ability to select certain things to go in or out of this membrane, is thanks to the facilitated diffusion and active transport, not to the other processes. Now, I've mentioned glycolipids before, I believe, and these are, as the name suggests, the bonding between carbohydrates and lipids. Carbohydrates, remember glyco for glycogen, carbohydrates, and lipids, well, it's in the name, and they're covalently bonded, okay? There's two types, glycoglycerolipids, the glycero for glycerol, okay, and the sphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphingosphing might seem very complex, but the only difference between them is what's in the middle, right? The glycerol or the sphingol. Now, they're amphipathic because their lipids are non-polar. This is why they're embedded in the layer. So the inside of the layer is hydrophobic, and because they have a hydrophobic region, they are embedded in the layer. But they also interact with the surface, with the extracellular environment, because their carbohydrate groups are polar and hydrophilic. It says hydrophobic there. Oh no. It should say hydrophilic. Yes. Just remember, lipids hydrophobic and carbohydrates hydrophilic. Now the glycoproteins are the covalent bonding of carbohydrates. In this time the oligosaccharides, which are short carbohydrate chains, to proteins. Okay. Now, functions of both of these glycolipids and glycoproteins: cell recognition, adhesion, and signaling. So, for example, you might have seen diagrams of red blood cells like this one. These spikes that they have on the end, the antigens, are actually made of these glycoproteins and glycolipids. Okay. So, for example, group A has A antigens, group B has B antigens, etc. Group AB has, antigens A and B, and group O has none of these antigens. So, as I said before, these glycoproteins and glycolipids are responsible for these antigens. See how topics are starting to interlink with each other. The glycolipids and glycoproteins make up what's called the glycocalyx, okay, which is a sticky layer on the surface of the membrane responsible for, as I said before, recognition. They are the antigens, okay, and there's also the adhesion to other molecules and signaling. You know, they'll receive signals from perhaps neurotransmitters or hormones. Now, moving on to the fluidity of the membrane. This depends on the amount of cholesterol in the membrane. We haven't looked at cholesterol yet, but when you practice drawing or at least labeling the cell membrane, you will need to remember that the amount of cholesterol impacts the fluidity. And also the length and the saturation, so whether it's monounsaturated or polyunsaturated when we're talking about the fatty acid tails. In other words, their nature, okay? Their nature is their length and saturation. You might be actually asked to label different components of the cell membrane, whether it's a multiple choice question or maybe even a long answer question. Mention a 7-mark question, mention all the components of the cell membrane, imagine. Anyways, so you need to be able to label it, right, and identify all the structures. So let's go through this one by one. First thing, integral protein, right here, okay? So this integral protein is a transmembrane protein. It goes across the membrane. We can see it has hydrophobic regions because it's interacting with the fatty acid tails, and hydrophilic regions because it's also interacting with the outside, the extracellular fluid. Transfer protein, also transmembrane. It's a channel protein and perhaps there's a carrier protein around here. Okay. Then we have the glycolipids, the glycoproteins. They make the what was that called? I swear I need to revise that as well. Well, the layer we just looked at. Then the peripheral protein, which only has a hydrophilic region, and that's why it's only on the surface. And the cholesterol, which we haven't looked at yet, it's within the membrane. Okay, so it's inside. I believe it can be outside as well, perhaps. And yes, let's go.